Let's get started. Hello, good morning, good afternoon, and good evening. Thank you all for being present for this second LinkedIn Live proposed by Netri. Just keep in mind that this live will be recorded and available in replay via Netri's platform. During the live, you can ask your question at any time directly on the chat, and do not hesitate to send us your question by email also. I am Jessica Rontard, R&D scientist at Netri for more than 2 years now, and very happy to host this webinar session whose objective is to present our technological solutions for growing 3D cell cultures in a reproducible way, thanks to microfluidic technology. Just a reminder, Netri is a French private company based in Lyon. At Netri, we design, manufacture, and sell standardized organoid on chips for application in neurology and other fields of application. We believe that organ on chips, that mean in vitro model using microfluidic technology, are appropriate to reduce the cost and ethical burden of animals models while accelerating drug development. Such systems relied on both the relevance of the human cells used with the cell cultures protocol adapted to the specificity of microfluidics and the ability to construct control physiological and relevant microfluidic architectures to the brain. It is important to note that organoid on chips offers the possibility to analyze the neural network thanks to MEA recording, as Netri devices are compatible with this approach. To do so, we are composed of multidisciplinary team with several expertise: a fabrication team to produce custom microfluidic devices, a biology team to validate that the technology does not interfere with the viability and functionality of the cells, a digital team to offer added value and create software specifically adapted to the device, and a team to support the need of our customers with a constant concern for the end-user experience. At Netri, we propose, of course, microfluidic products, but also applications focused, for example, on neurological troubles, such as Alzheimer's disease and axonal regeneration. And also, we propose a R&D service and specific capabilities. Today, we will focus on one of microfluidic devices' capabilities for growing 3D cell culture. I will start this presentation with a short introduction of the preliminary data obtained, and then we will continue with a direct testimonial of one of our partners. We will focus now in the following presentation on cerebral organoid, but all the technological solutions presented here are applicable to other types of 3D cell cultures. So do not hesitate to contact us for more information about this point. Neurology is one of the failure areas in the drug development pipeline. This high drug attrition rate suggests the limitation of current experimental tools leading to clinical application, and the gap between in vitro models, rodent models, and human situation. Modeling the brain remains an elusive challenge because of its inherent complexity. This complexity begins at the cellular level. The human brain is composed of cellular heterogeneity, composed of neuronal and non-neuronal cells, in a complex 3D neural network organization. Cerebral organoids are emerging as alternative methods to 2D model studies and as a powerful tool to modelize complex biological process thanks to the ability to recapitulate many features of the human brain. Despite the major advance of the cerebral organoid research, there are still some limitations to their transfer and huge adoption by scientists for neurological disease modeling and drug testing. Indeed, we can see the lack of macroglia cells, vasculature, the depth in the center of the organoid, as well as the difficulty to obtain reproducible cerebral organoids from one laboratory to another, and the need of high-throughput analysis methods such as non-invasive and scalable. The goal of the NETRI's Organoid-on-Chip program is to propose a microfluidic devices compatible with organoid generation protocol and allowing to enhance reproducibility and quality of the organoids. We have designed a 3D deposition chamber allowing the growth of 3D cell culture up to 4 millimeters of diameter, called nest cube. All NETRI's devices are made with Pdms, and standardized design and fabrication steps allow the generation of reproducible 3D cultures. The devices is aligned to 19/6 well plate format and easy to use without pump equipment needed. The nest cube is also compatible with confocal microscopy, as you can see on this immunofluorescent picture. There are several methods for generating cerebral organoids, resulting in different types of neural organoids. In this project, we have chosen Lancaster protocol with embryoid bodies embedding in matrigel scaffold. To do so, the first step was to set up a protocol to polymerize matrigel directly in the nest cube, because we want to introduce the cerebral organoid as soon as possible to enhance reproducibility. Here you can see 2 examples of different eggs of scaffold thanks to fluorescent colloids in the nest cube. We then developed a protocol in collaboration with a French academic team to introduce the embryoid bodies 2 days after their generation into the microfluidic devices. Preliminary data allowed us to demonstrate cell viability for 60 days in the nest cube. After we cover it and fix it, we perform immunofluorescent staining and observe, as you can see here, the presence of MAP2 positive cells and GFP positive cells. All this work was being done in collaboration with Celtex, a French academic team specialized in the culture of cerebral organoids, and in particular with Héloïse Castiglione, who prepared a short video for us. Thanks again, Héloïse, for this presentation.
Hello, I'm Héloïse Castiglione. I have just finished my engineering studies at Subbiotech, an engineering school in biotechnology, and my end of studies internship was a collaborative project between NETRI and Celtex, which is a laboratory specialized in cerebral organoids. And the internship consisted in developing and optimizing a human brain organoid-on-chip system by culturing cerebral organoids within a NETRI's microfluidic device. In fact, brain organoids are 3D self-organized cellular aggregates that model in vitro the human brain, but they are currently not completely adapted to an industrial transfer and for high-throughput analysis in preclinical applications. So the objective of the internship was to improve the culture conditions of the brain organoids thanks to the advantages offered by microfluidics to improve viability, maturation, and reproducibility of the generated organoids, and with a long-term purpose of developing a predictive and standardized in vitro model of the human brain that answers the needs of the pharmaceutical industry. And during the internship, we collaborated and exchanged a lot between NETRI and Celtex to adapt the cerebral organoid generation protocol to the NETRI's microfluidic device. And when I encountered some difficulties, my NETRI supervisors helped me to find solutions. And finally, cerebral organoids had been cultured in the device up to 60 days and were characterized, and they globally displayed the expected characteristics in terms of morphology, growth, and cellular populations. And these results were presented in posters at the RUX Congress in July and more recently at the FSSCR Congress in October. But there is still work to be done to further characterize and to improve this brain organoid-on-chip platform. So we are currently pursuing this collaborative project, and I will begin very soon my CIF PhD between NETRI and Celtex. And recently we have submitted together a review about recent advances of brain organoid-on-chip systems and the main challenges that remain to be overcome for the preclinical applications.
We really thank Eloise and Celtex for this presentation. In conclusion, combining cerebral organoids with microfluidic technology, referred to brain organoids on chip, allow to model the macroenvironment of several neuronal cells types in 3D. The cerebral organoids maintained in Nest Cube are available until day 60, with the presence of neurons, astrocytes, and proliferative cells. The next step is to continue the characterization of organoids and analyze the advantage of macrofluidic technology to reduce, for example, the depth in the center of the organoid. In perspective, we want to design a new devices to co-culture several cell types with organoid to model, for example, the blood-brain barrier. The next webinar will take place next week, and I will be excited to talk about our technology solutions to study synaptic transmission between human cells and neurons. Thank you very much for your attention and your time. Let's see if there are any questions. I think we have time for some questions. As a reminder, you can post your question via the chat, or send us an email for further contact. No question for the moment. Hi, thank you very much for your comments. If you have any question, please. Well, the first question is, can you list down many challenges in brain organoids on chips? Thank you for this question. There is a lot of challenge to culture cerebral organoids on chip. The first one is to find the good protocols and the good timing to seed the cerebral organoid in the devices. Because as I mentioned at the beginning, we want to enhance the organoid reproducibility. So we have to find the good timing of the seeding. The second challenge is, for example, to set up the matrigel scaffold protocol in the devices. What we can also say, it's to also analyze the organoid. We have found a good way to recover the organoid and to fix them, to perform, for example, immunofluorescence staining. So it's also another challenge. Another question? We have time for one other question if you want. Okay. Well, another question. How do you recover the organoid from inside the Pdms devices? Well, we have found a good way to recover it. If we want more detail, please send us an email and we can provide us the protocol for that, for sure. Another question, how difficult it is to measure electrical activity of chip-based brain organoid? Thank you very much for this question. We really want to find the good solution to propose and to to perform MEA recording. It's really a challenge. We have found some solution. It is in progress for the moment, but yes, it's really the next step to find the good way to recording organoids on the chip. Okay, no more question? Thank you very much for your attention. As I mentioned at the beginning, this live will be on replay via NetWith platform, and see you next week for the next webinar. Bye-bye, everyone. Thanks.